lithotropic, and what these guys have found is that they're not using light from the sun. Their only source of light is geothermal radiation, which is a wavelength of light that you wouldn't even really call light because it's not light that you can see with your eyes. It's radiation that can be used as an energy source. And so these bacteria are living down there conducting photosynthesis obligately, but they're not getting light from the sun. And so you can imagine that when that was first discovered, nerdy microbiologists like myself associated that with the possibility of life living in other parts of the universe or the solar system, right? Because they say that on some of the moons of the big planets that are farther out, there's frozen oceans, and there's probably no chance of light penetrating down there. So the question is, how could you ever have primary production? So primary production is not just driven by phototrophs using the sun. It can also be driven by phototrophs using geothermal radiation. And primary production is also driven by CO2 fixation in situations where the cells aren't using the sun at all. So when you think about primary production in ecology, what is the definition of primary production? Where would you have learned about primary production in ecology? Something like Bio 120, right, Ben? You've taken a whole bunch of ecology classes, right?
Yeah. So it'd be like Bio 120 is what I remember.
What is primary production?
So primary production, if I remember correctly, is like your grasses that grow in the ecosystem, is your base formation of the ecosystem, sort of your...
It's the base. It's like the bottom of that pyramid that you always saw. It's at the bottom. And why is it at the bottom? What is going on by the primary producers?
The first ones to get energy.
Right. They're the ones that are fixing the CO2 and making the food and collecting the energy for everything else that eats it, right?
Yeah.
And so we typically think of those as phototrophs, but it's really actually anything that's autotrophic. And so a huge amount of organisms in the world are primary producers, but they are non-phototrophic. They're autotrophic. And that just means that they use CO2 as their electron source. So chemolithoautotrophs, and of course that takes a lot of energy. So chemolithoautotrophs are getting their electrons from an inorganic electron donor, lithos meaning rock, and that is not a particularly favorable way to do it compared to using glucose. This table, the only reason I have this up here is for comparison. You don't have to memorize these numbers. This shows the delta G for a bunch of inorganic electron donors, and down at the bottom they say the delta G for complete oxidation of glucose to CO2 is minus 686 kcals per mole. 686 compared to 65. So for example, glucose, complete oxidation of glucose gets you 10 times more energy than this. And this is a pretty good one. Thiosulfate's pretty good too. But the point is that since they're not able to use sugar for energy, they have to use a whole bunch of these different inorganic electron donors to get enough electrons to be able to do their business, right? So they go through loads and loads of this stuff, and it makes them really important in the environment because they're responsible for moving a whole bunch of mass through the biosphere in terms of biogeochemical cycling. So when we talk about the nitrogen cycle later, we'll talk about these and think back about how much ammonium and nitrate must have to be used by the organisms in order for them to do all the stuff that the microbes do, compared to, in a molar level, compared to glucose, for example. So we're going to talk about two different groups today in terms of chemolithoautotrophs that you haven't probably thought about before, and one of them is the organisms that oxidize sulfate, or do sulfur metabolism. So electrons from inorganics. An example, sulfite. So sulfite, this, you guys probably can't read that writing real well. This is sulfite. And it's got some electrons in it, right? If you take electrons away, the most reduced form of sulfur would be sulfide, right? Hydrogen sulfide, that would be the most reduced. That would have the most electrons to give up. This doesn't have that many, but it has an electron to give up. It can be eventually converted into sulfate. And there's two different ways that this can be used as an electron acceptor. One of them is direct oxidation to an electron transport chain. So what would that look like? If you were to draw a picture of that, it would just be like every other old picture of electron transport chain. The proteins in there would be different. And for if you were a biochemist, that would probably drive you wild and you would learn all about every little reaction in there. But let me see. I'm just talking about, I want to go all the way back to my slides. Something like this, where electrons get donated up here, not way down here like you would usually have with NADH, but up here, electrons would be donated to some carrier, and that would drive proton pumps and make a proton gradient. So we're talking about organisms that aren't doing that. They don't have loads and loads of NADH because they aren't growing on stuff that has a huge amount of electrons to give up. So that's one way that they can do it. Another one is a different kind of substrate level phosphorylation that is shown on this slide right here. Substrate level phosphorylation with this intermediate APS. So what's going on here? This is the bottom part of it that we're talking about here. This is the part I mentioned right here. Whoops. This is the top part where you could have regular direct oxidation to an electron transport chain, because these electrons could go to an electron transport chain to make a proton motive force. Down here, you have formation of ATP right here. That would be substrate level phosphorylation. And here's what happens. It uses an unusual intermediate called adenosine 5'-phosphosulfate. It's sort of like ADP, only instead of a second phosphate here, ADP is diphosphate. This has a phosphate and a sulfate here. So what happens is, to start with, sulfite reacts with two AMPs, adenosine monophosphate, to make two of these, two APSs, and you get 4 electrons out of it. So those 4 electrons can also be used for the electron transport chain, because this is essentially reducing power. Then you have these two APSs react with phosphate, inorganic phosphate, to make two ADPs, and you get sulfate back. This is not sulfite, but sulfate. And then two ADPs interact with AMP, or get phosphorylated to give you ATP by substrate level phosphorylation. So there's lots of places in the world where substrate level phosphorylation occurs. This is special and unique because it involves this intermediate, adenosine 5'-phosphosulfate. So with this, you get both ATP and reducing power. But clearly an organism that's doing so this is happening in your Winogradsky columns. This is the kind of thing that's happening in the Winogradsky column down where it's dark and anaerobic. It takes loads and loads of sulfate and sulfide and sulfur to get the cell enough energy to be able to function. And so you go through large amounts of molar amounts in order for a cell to have enough energy. So that's one example of chemolithotrophy and the kind of energy extraction that a cell would use. There's another one that I want you guys to look at because it includes some other concerns. This is using nitrite as an electron source. Nitrite as an electron source. So what's happening here? This shows, the book tells us what, or this isn't Paracoccus denitrificans. They use that one a couple of other times. This is Nitrobacter. We're going to talk about Nitrobacter again when we talk about the nitrogen cycle. But it shows the electrons. It shows the electron source right here. This is nitrite. NO2 minus is nitrite. It gives up one electron, becomes nitrate. And like we said on that table that we first looked at, you don't get a lot of energy from this. But the electrons are used to generate a proton gradient. That's happening right over here. And what's not shown is that you'd, of course, have an ATPase here, making ATP. That's where they can get their ATP. And this fits the model that you thought about before. The protons get pumped out, you have a whole bunch, it comes through and makes ATP. But these organisms, because we're talking about chemolithoautotrophs, they also have to have a lot of energy that they're using for fixing their CO2. So you guys learned about CO2 fixation probably in Bio 121. And in that context, you were probably talking about the Calvin cycle. How many of you guys remember anything about that? It kind of comes back to haunt you. Here's the figure in your book about the Calvin cycle. This is on page 270. Figure 12.4. So you don't have to learn the whole Calvin cycle. I'm just showing it so you guys have a little reminder about what's happening. This is one of the ways that bacteria that are autotrophs, non-phototrophic bacteria that are fixing CO2, do it. They take CO2 and add the carbon from that to ribulose 1,5-bisphosphate to make 3-phosphoglycerate, and then they go through here, bring it down here. Eventually, they make the biosynthetic products that they need. All of the carbon, all the fixed carbon they use for all their amino acids and everything else has to come this way, and they use NADPH for energy. And so when we were talking about energy before, and NADPH, we were talking about the Entner-Deuteroff pathway. We talked about NADH from Krebs cycle and glycolysis and everything else. If these cells aren't using those, they need a whole bunch of NADH in order to fix the carbon. And they don't have the same source of NADH that the other organisms have. So they're using this side of the cycle over here and reverse electron flow to make their NADH for biosynthesis. So they're not getting loads and loads of electrons from organic material like glucose. They're getting tiny, pathetic little amounts of electrons from nitrite. And so they have to take the proton motive force that they made and also use it to make ATP, but they also have to use it over here to drive the formation of NADH. The NADH that they're going to use for biosynthesis. They're not going to use this NADH to make a proton gradient like the chemoheterotrophs do. They're going to just have it around because they're going to have to use all that energy to make their every organic molecule in the cell, that they're bringing in one carbon at a time from CO2. So these guys are doing two different things with this electron transport chain. One is backwards to our thinking because they're generating NADH. It could be NAD plus too. And they're also making ATP on this side using proton motive force. So two roles for proton motive force. One is ATP production. Two is NAD or NADPH production. So that's what it means to the bacteria. What it means to us is that microbes that are doing this, and they're often soil bacteria, are using tons and tons of nitrite. And they get their nitrate sometimes from ammonium. This could be happening with ammonium too. Or they could be getting it by reducing nitrate. And all of that, for us in this part of the world, has agricultural significance because this is a way that nitrogen in the soil gets used by bacteria and may not be available for the corn and the soybeans that we're trying to give it to. Uses huge amounts. Questions? So these are two things that are probably totally new in terms of have you been exposed to them. New to you, right? You didn't learn about these in other classes. This is a case where citrate phosphorylation involves a weird intermediate, this, and that is formed from the sulfur that comes from sulfite. This is weird because the cells are using their proton motive force for another thing that we haven't talked about before. So that's the fourth thing we've talked about proton motive force being used for. Transport, flagellar rotation, ATP production, and generation of reducing power. Questions about those? I see everything from, oh my God, I have no idea what's going on, to this is so obvious I can't believe you just spent 20 minutes talking about it. Makes my job kind of hard. Does anybody have any questions about the big picture? So again, this is big picture stuff. We're not going to learn about what is cytochrome C and all of the details in there. This is more about the world than it is about individual enzymes. Okay, let's talk about... So I updated Moodle, if you guys haven't seen it. I put stuff on there about what's going to be next. I also put stuff back on there about Chapter 7, which we dumped off before the test, and now it's back coming up on Friday. I just moved the same page numbers and the same independent learning down to this week from Moodle. So we'll talk about that on Friday. Today, let's talk about a couple of topics in anabolism.
Dr. Eglin?
Yeah.
That last sheet that you had up?
Yeah.
If it has the ability to go through the electron transport chain, why does it want to make NADH out of it?
If it has the ability to do the electron transport chain, if it has the ability to do this,
Why doesn't it just use it all to make ATP? Why would it need?
it's because it's using NADH in places that it needs to fix carbon. It's using it for anabolism. So the Calvin cycle is one example of that. The other example is reverse electron flow using, let me show you this, using the TCA cycle. So this is the the Krebs cycle, or the TCA cycle, aka the TCA cycle. And here it's shown functioning anabolically. This is perfect timing for the question. Remember when we talked at the beginning about amphibolic pathways, and we said that the pathways can be used for catabolism, and you get the electrons out and make energy, or they can be used the opposite direction, and you can use energy to make the pathway intermediates. This is a case where energy is used here in the form of NADPH, NADH, there should be another one in here somewhere, investment of energy here to fix CO2. So here's CO2 right here. In an organism that's fixing CO2 photolithotropically or chemolithotropically, they have to use either the Calvin cycle or they use the TCA cycle in reverse. So these guys aren't getting any NADH out of their Krebs cycle or their TCA cycle. They're actually consuming it because they're fixing CO2 here. CO2 is going in here and here, and it's going backwards, and energy has to be invested. So to your question of why don't they just, let's see, I don't remember what your question was. Why don't they just consume NADH?
Because generally we consume NADH.
That's right. They don't have enough NADH to consume because they're using it to fix their carbon. Or NADPH. We're using them kind of interchangeably here. Because they're either using this to do reverse TCA cycle or to use the Calvin cycle. Very good question. And good segue into anabolism. So, speaking of that, we talked before about how we're not going to memorize how much ATP you get out of every single molecule of glucose because it's not a realistic scenario that you would be able to have access to that because of the fact that the cells are using some of the carbon going through there for anabolism. Some of these intermediates are being taken out of the pathway along the way, and so you don't actually have access to them. That certainly would be the case if you have a CO2 fixing organism. You'd have CO2 going in to make all of this stuff, which then would be used for making amino acids, right? So we're not going to talk about all the reactions for making amino acids. We're just going to talk about a couple of unique things that happen with anabolism in prokaryotic organisms. 2 topics on anabolism. If you take biochem or some other class, you'll learn a whole bunch about different topics on anabolism. The one that is especially important is nitrogen fixation. So nitrogen fixation is the reduction of atmospheric nitrogen to ammonia. Atmospheric nitrogen is N2, right? Molecular nitrogen to ammonia. So here's a common misconception that people have misunderstood before. If you have, let's draw it out like this. It's got a triple bond, right? It's super stable. If you have this serving as being reduced, let's put electrons. I'm going to write the word because there's many. We'll talk about it later. Electrons. If electrons are going into this, why does this fit into anabolism? And we talked before about how nitrate, for example, can be a terminal electron acceptor, or you can have all kinds of inorganic terminal electron acceptors under anaerobic respiration. Why does this not result in, why cannot this not be thought of as a terminal electron acceptor in respiration? The results here, I should have drawn it a little bit differently. Let me go back to a normal way of drawing. Takes 8 electrons, gives you 2 NH3s. The reason that this is thought of anabolism and not just anaerobic respiration is that you're using it to build something. These ammonias are going to be used for making amino acids. And so this fits into the category of anabolism. And in fact, this isn't part of energy generation. This takes tons of energy. Let's write out the whole balanced equation. The whole balanced equation involves tons of energy, 8 electrons plus 16 ATP. So they're not making ATP, they're costing ATP to get 2 measly aminos, enough to make 2 amino acids. That's a very expensive process. You also get hydrogen. Of course you get, this is not the interesting part, the 16 ADP and the pyrophosphate that goes with it, right? The point here is it's extremely energy expensive. And this is not a way that they make energy using this as a terminal electron acceptor. This is a way that they spend energy to make this ammonia for amino acids. So this is a process that only happens in prokaryotic organisms. Which is super important for us in terms of agriculture. We say that, farmers rotate their crops, or at least they used to. Do they still rotate crops? Yeah. Rotate their corn and their beans because the beans help put nitrogen back into the soil. It's not really the bean plants themselves, of course. It's the bacteria that live in the symbiotic bacteria that live in the nodules on the bean plants. And so all of the fertility in terms of nitrogen in soil is either catalyzed by bacteria that live in the soil or is put on artificially so that we can afford to grow enough corn to feed the whole world. So we're going to talk about a little more detail about that next time, but first I want to give you guys back your exams. So we're going to have to stop here. We've got 10 minutes. Okay, everybody listen before I hand these back. Once I hand these back, I know I will have no control. The average was 80%. There were only 71 points on the exam. It was a short, cheap exam. The correct score is written under 71 on the second page. So if you see you got a 50, that's 50 points, not 50%. So don't freak out about that. I need to have all the exams back. The correct answers should be written in, but we're going to go through them and talk about them here.
Heavy breathing. I'm so nervous.
Did I tell you the average was 80?
Strangely, that is almost exactly for making up workouts. How do I sign in for that? Do I just the one that I missed, and then just sign it, and then just say like made up this day or anything? Okay. Okay. Sounds good. Okay. So it doesn't. So if we go and make it up, it doesn't have to be during the workout time. We'll just go. Okay. That's good.
I mean, it's like kind of like during the meal time, like if you can, like, I think it is for Saturday.
Okay. Okay. Yeah.
Yeah.
Lisa? Lindsay. Joe. Is Brooklyn out there? No, he's with. He's with. Okay, does anybody have any questions about the multiple choice? The correct answer should be written in on the multiple choice. Questions about those? Okay, fill in the blank. So these got a little bit messy in some places because I didn't make my key so all-encompassing that the TAs necessarily could read my mind. So I had to go back on some of those. Which part of the cell wall is responsible for differences in the gram stain? Explain your answer. The answer is peptidoglycan, and the reason it's responsible is because in gram-positive there's enough peptidoglycan to trap the crystal violet, making it stay purple when you stain it. 2, give one difference between the bacterial plasma membrane and the plasma membrane of archaea, which I spelled wrong. I see I left out the R. Archaea have rings and branches. They don't just have a straight hydrocarbon chain. They also are built, their lipids are built with 5-carbon isoprene units rather than 2-carbon acetate units. Also, in archaea, sometimes they don't have a bilayer, they have a fused monolayer with phosphates on each end, which is kind of weird. Difference between the cell wall in bacteria and in archaea. Spelled it wrong 2 times in a row, I see. Bacteria have peptidoglycan and archaea don't. They have something called pseudomurine. They also have lots of different protein layers and there's a lot of variety in what they can have, but they don't have peptidoglycan. 4, what's the major way that replication of a retrovirus differs from other RNA viruses? So it's about replication. How does a replication differ? So you had to address that. Retroviruses form a DNA intermediate that enters the genome as a provirus. So the replication relies on a DNA intermediate that is in the host cell's genome. So your answer had something to do with that. 5, the major factor controlling tissue and host specificity include the viral structure involved. The answer, to get full credit, you had to say something about the viral spike matching the host cell receptor. So in order for a virus to get into a cell, it has to contact, and it can't contact just any cell. It has to be the appropriate cell with the appropriate receptor. 6 types of viral proteins that would be expressed late: capsid proteins, protomers, spike proteins, proteins that get packed inside the virion. So it would not be their early genes or their intermediate genes. It would be stuff that is used for making the virion. 7, how is negative single-stranded RNA used in viral replication? It's used as a template to make positive single-stranded RNA. So that is a case where I went back and gave you, some of you guys might have written something about mRNA, and that you should have gotten credit back for that. 8, one characteristic typical of bacteria is their small size. How is it related to the surface area to volume ratio? The way it's related is that as the size of the organism increases, the ratio of surface area to volume decreases, making it less favorable for the bacteria because they have to, remember, transport every single thing across their membrane using transporters, and so they don't do endocytosis. So if the bacterium gets too big, they don't have enough access. The endosymbiotic hypothesis is the idea that eukaryotic cells, organelles, evolved from prokaryotes that were taken up by them. And there are lots of examples. In fact, we know enough about them to know what bacteria they're most closely related to. So their 16S rRNA is an example of a piece of evidence. They have circular chromosomes. There are also extant examples of obligate endosymbionts in which eukaryotic cells have a prokaryote living in them that they require. Number 10, what is the energy source for movement of the bacterial flagella? The answer is proton gradient, or if you wrote H plus gradient or proton motive force, those are all the same thing and they're all correct. But the hydrogen is not the correct answer. 11, peptidoglycan side chains are characterized by the presence of D-amino acids. They confer resistance to proteases, which act upon L-amino acids. 2 roles of lipoteichoic acid. They attach the peptidoglycan to the plasma membrane, making it stronger, and they attract magnesium ions. 13, what is the difference between a microbial species and a strain? A strain consists of descendants from a single pure culture. A species is a collection of strains with common properties. So the definition of a species used in all the rest of biology has to do with interbreeding pairs of organisms. But since bacteria don't breed to reproduce, that doesn't work for them. And so a species includes a whole bunch of different strains. In fact, you can make your own strains by mutagenizing them or selecting for differences. Purpose of bacterial endospores, survival of harsh conditions. 13, 14, and 15. Discovery of bacteria using a microscope was Van Leeuwenhoek in the 1600s. 14, disapproval of spontaneous generation was Pasteur in the 1860s. And if you put development of techniques for sterile technique, you should get credit for that too. Pasteur is also credited with that. 15 is recognition that bacteria cause disease was Koch in the 1880s.
What was the total on that page?
What was that?
What was the total? on that
The
page?
total on that page is 23. The 13, 14, and 15, which I see now should have been labeled 15, 16, and 17, are worth 1 point apiece. The others are worth 2 points per blank. As far as adding up your points, the first page is worth 12 points total. The second page is worth 18 total. The third page is worth 23. The fourth page, which you're going to do now, is 10. And page 5 is 8. Okay. Other questions? Page 4. So you know what? When people miss points, in general, it was because of a lack of specificity or not enough detail. Name 2 specific examples of inclusions found in prokaryotes and state their functions. So examples would be polybeta hydroxybutyrate granules for use in storage, or glycogen for use in storage, magnetosomes, not ribosomes. Ribosomes are never the right answer. So it was worth 4 points total, 1 for naming it properly and 1 for saying what it does, and you had to have 2 of them. Number 4, that was a joy to grade. Or number 2 was worth 4 points. To get full credit, you had to reference a series of runs and tumbles. You had to say that cells spend more time running or tumble less frequently when they're going in a favorable direction, because that's the whole basis of how it works. The question asks what part is random, and the part that's random is the direction. The direction is random, and in a uniform environment, you had to address what would happen if a cell was in a uniform environment. And we actually, based on the information you had so far, it would have been pretty hard for you to feel confident about that, but if you just put something that is a reasonable guess, like they would create an environment and then they'd move towards a concentration, or they would run at an equal amount all the time, that was fine. So that was worth 4 total. 3, besides causing mutation, how could a virus result in malignancy or cancer? They carry oncogenes, so they could deliver oncogenes to a cell. They could change the regulation of proto-oncogenes. They could deliver genes encoding products that turn off tumor suppressors. That was worth 2 points. The last one, draw and label the diagram. Remember how I told you guys how to draw stuff? Some of these were beautiful. In fact, I took a picture on my phone with one. My kid said, "Dad, why are you taking pictures?" Here's a beauty. So it has indication of all the parts. The outer membrane's out here. The labeling's got some issues. It shows different parts and connections in the peptidoglycan. Here's the plasma membrane. So you had to have those 3 parts diagrammed in a way that gave some indication that you knew what they were made out of or their structure. And then you had to have 2 parts that were virulence factors or somehow related to disease. So the O antigen is important because that is antigenic and our body responds to it. The LPS is important because it is endotoxin. The peptidoglycan is important because lots of antibiotics target peptidoglycan. The porins on the outside of the outer membrane are important because they're restrictive, and organisms that have mutations in those sometimes become antibiotic resistant if it increases the restriction. All right, we're almost at student presentations. Biofilm, growth on a surface. Bioluminescence and dinoflagellates is for scare predators away. 3, photorhabdous luminescence is found in nematodes, which are eaten by insects, and then the insects explode. You didn't have to have all that. This page is worth 8. Go ahead, if you haven't, add up the total. Make sure it matches that on the second page. I have to have them back. Average was 80%. My average this year is exactly the same as my average last year. Oh, the test wasn't exactly the same, but...
Did you good?
Yeah.
Did you do good?
I was expecting like a.
Okay, so you have that in the
Yeah?
context of...
So I actually did pretty decent. I didn't go bad.
And that's what I was like,
Enough.
okay, as long as I get